A population can be defined as a localized group of individuals belonging to the same species. Sometimes, few populations shows distinct boundaries like population of white-footed mouse in Sanford Natural Area. However, in other cases, population did not have demarcated boundaries, therefore it is randomly set by the convenience of the investigators, for instance, eastern chipmunks around Holmes Hall. The next step after defining the boundaries is the determination of a population size. It can be done by either counting total number of individuals from a given smaller sample area which can be extrapolated to the set boundaries, or it can be done by capture-recapture method. The population biology is mainly concerned with the factors which can alter the population size with time. The important factors are mainly natality, mortality, growth of individual and reproductive success. The strength of population ecology lies in its quantitative nature. For example, what will happen if survival rate of bald eagle is decreased by 2% per year? Or if survival of juvenile salmon is increased by 0.5%, how many adult salmons would reach to their maturity? These simple questions can be answered by applying simple mathematics in the population ecology. In this module we will understand the quantitative background for population ecology. 3. Semmelpris versus Iteroporus life cycles. Some species, e.g., most invertebrates, in their lifetime have only one reproductive event and after that they die. The efforts of these species in the reproductive events spend enormous energy in order to produce large number of eggs, whereas, other species, e.g., most birds and mammals live to perform several reproductive events in their lives. There is a great amount of variation in these two categories. For instance, some species of Semmelpris organisms have overlapping generations of young ones. As a result, there may be one year or 2-year-old individuals can be found in the population. The common form of Semmelparity found in the insects living in temperate regions is an annual species. In these conditions, the insects survive as an egg or larval resting stage during overwinters until spring, and after that in the summer months they grows and matures into the adult which perform reproductive events. After mating, these adults lay eggs which survive in the same dormant stage in adverse condition throughout the winter months. However, several other Semmelpris species can produce several generations each season of summer. It is now easy to understand that importance of reproductive event frequencies, the young ones produced in each reproductive event, and generation time that can greatly influence how fast a population can grow. 4. Life tables. Mortality is one of the important parameters that derive changes in the population. There are several kinds of questions such as, is mortality more among young organism? Does an old-aged individual have higher mortality than younger organism? We can answer these kinds of questions by constructing a life table. Life table was built up by those human demographers who were working for life insurance companies and were interested to estimate the life expectancy of the people. Plants and animal populations may be composed of several types of individuals, and in any given analysis a demographer may group them together or may keep them separate. 4. A life table can be explained as an age-specific summary of mortality rates that is operating on a cohort of individuals. A cohort may consist of either the entire population, or it may include only the males or females individuals that were born in a given period of time. The life table is most frequently used by the life insurance companies to find out the probability of survivorships of human populations for deciding the premium, whereas ecologists uses life table to study the natural population. The life table is most frequently used by the life insurance companies to find out the probability of survivorships of human populations for deciding the premium, whereas ecologists uses life table to study the natural populations. Raymond Pearl, 1921, Figure 1, first used life table for the population of Drosophila reared in the laboratory. Figure point 1. Raymond Pearl. Generally, it is very difficult to construct life tables of non-human populations living in the wild conditions. The life table construction is a very simple method to keep track of birth rate, death rate, and reproductive events in the given population. The following types of data can be used to construct the ecological life table. Laboratory animals. The life tables of these animals of known age can be constructed by directly observing the individuals which are dying at regular intervals, and the observation will be continued till all individuals in a population died. Survivorship directly observed. The survival of large cohort can be studied by following at regular intervals till their existence. Connell, 1961, studied mortality of the barnacle 5. Cathamalus stellatus, grew on the rocks during autumn season in Scotland. The mortality of these barnacles was observed at regular interval, and the data was used to construct the life table. He did several experiments in which he removed one competing barnacle Balanus balanoides from some rocks, and then he counted the number of Cathamalus surviving once in a month. Age structure directly observed. This type of data can be used to construct the static life table, particularly of trees, birds, fishes, and many more. The information regarding age structure can be acquired by observing the annular rings in the horns of sheep, growth rings in the cementum of teeth in case of ungulates, as well as carnivores, and annular rings present on the fish scales. For example, in a pond, we can get a sample of fish which can be used to determine the age of each fish simply by analyzing the number of the annular rings present on their scales. Similarly, age of the tree can be determined by counting the tree rings. However, these types of data are not always preferred for making life tables. Age at death. This type of data can be used to construct the static life table. We assume that population size is constant over time and birth rate, death rate of population at each age interval remains constant. Sinclair, 1977, constructs the life table of African buffalo, Cincerus Kaffir, by collecting 584 skulls that died due to natural causes. After that, he classified these skulls on the basis of sex and age. The age of the skull was determined by analyzing the annular rings on the horns. The estimation of age of young skull was difficult because they were fragile and more susceptible to carnivores and damage by environment. He observed the losses in first 2 years of life by directly observing the herd and obtained the mortality data. 5. Types of life tables. The life tables for human populations can be formed by analyzing the data of birth and death recorded by municipal committees of different states as well as by life insurance companies. Life tables can be classified into following 3 types. 6. 5.1 Horizontal life table. It is also known as cohort or dynamic life table. This type of life table is constructed by observing the survival and reproduction of all individuals of a cohort from the time of birth to the death of all members of the population. A cohort is considered as the group of all individuals born or recruited into a population during a given period of time. For example, a cohort can be start with 1,000 individuals and observation of number of dying individuals can be done at regular intervals till all the individuals in a population is exhausted. It is usually related to those species which have short lifespan and their generations are discrete. 5.2 Vertical life table. It is also considered as time-specific life table or static life table. The vertical life table can be constructed by recording the total number of individuals surviving at each age group in a given population and their reproductive output. This life table is based on the assumption that each age class is sampled in proportion to its numbers in a population. It also assumes that the population is stable and death rates and birth rates in a population are constant. 5.3 Dynamic composite life table. The construction of this type of life table is similar as cohort life table. However, in this case, the cohort is a composite of a number of individuals that are marked over a period of years rather than at the time of birth only. The dynamic composite life table and static life tables are considered as less accurate, whereas cohort life table provides a reasonable idea about average conditions of the populations. The life tables are very useful in comparing the trends of life history within a population and also in between different populations. The cohort or static life tables which classify individuals of a population by age groups are called as age-based life tables. In these types of life tables, individuals who survived for less than 1 year are assigned 0 age and those individuals in a population which lived for 1 or more year but less than 2 years are allotted 1 year age and so on. However, size-based life tables and stage-based life tables usually classify individuals by their size or stage of developmental instead of age. Generally, these types of life tables are more useful and more 7. Practical for understanding those organisms which cannot be differentiated by their age as well as their ecological roles or chiefly depends on their size or stage rather than age. 6. Components of a life table. 1. First column X in the life table provides age intervals of the individuals in time units suitable according to the species. 2. Second column NX gives number of individuals surviving at that particular age interval. 3. Third column LX gives proportion surviving. It can start with 1,000 individuals that enter in the first age group and gives the remaining number at the beginning of each subsequent age interval. It is calculated by the formula LX equals LX1 NX1 X NX24. Fourth column DX represents number of individuals dying at each age interval from the initial group of 1,000 individuals. It is calculated by DX equals LX plus 1. 5. Fifth column EX denotes life expectancy. It determines the total expected survival of an individual of a given age beyond its present age. It can be calculated by following 3 steps. A. LX is average number of individuals alive. Calculate the proportion of survivors at the midpoint of each time interval. LX equals LX plus 1 2. B. TX is total lifetime remaining for all the individuals attaining the age. Sum all the LX values from the age of interest up to the oldest age. TX 9. 7. Construction of cohort life table. To construct a cohort life table, for example, individuals born in the United States during the year 1900, the survival of these individuals were recorded from the beginning of 1901, 1902, 1903, 1904 and so on until all individuals were die. This record is called the survivorship schedule. Mostly, life tables calculate only females and their female offspring for those animals in which there are males and females and both of them are equal in numbers of each age. Most plants, hermaphrodite animals and other organisms in which there is no sexual dimorphism, life table calculations may have to be adjusted. The construction of life table for this simple life history can be done by counting the population size at each life stage. The first column X denotes the age class and second column NX specify the number of individuals survived at the beginning of each age. These data can be used to calculate several other life history parameters, such as the proportion surviving at each life stage Lx, proportion of individuals dying at each age dx, and AGA-specific mortality rate Qx dying at each age, which is helpful in locating the points where mortality is most intense. Example point 1. Cohort life table of song sparrow on Mondarte Island, British Columbia. Source: Smith 1988. Age in YRS. X. Observed number. Birds alive each year. (10) 420.01710. For instance, by making the age for constructing life table, the first step is to decide age interval in which data can be placed. For instance, human and tree can be grouped at interval of 5 years. For birds or deer population, interval can be of 1 year, and for rodents, field mice, or annual plants, the interval can be of 1 month. By making the age interval shorter, we can analyze the mortality scenario in more details. The rate of mortality Qx is expressed as a rate for the time interval between successive census stages of the life table. For example, in the table, 78% of song sparrow died before reaching to age group 1. Example 2. A cohort life table for Phlox drummondii. After Leverick and Levin, 1979. Age in interval days X. Number. O 63996. When sheep was either killed. Adolph Murray collected many horns. When the age of the sheep was. Life table of Alaskan population of Dall Mountain sheep is the most famous life table. The age of the sheep was estimated by the rings of the horn. Older the sheep, more bony rings. When sheep was either killed by wolf or died due to some other reason, horn remains preserved in the soil. Adolph Murray collected many horns, which provided exhaustive data of sheep age which died in an environment due to natural hazards including wolf predation. Example 4. Life table data of Ballin. 6.5. 8. Several organisms. 8. Construction of static life table. Several organisms have complex life histories, especially for those organisms which are mobile and have higher longevity. Therefore, it is very difficult to follow all the members of a population throughout their survival. Therefore, there is another less accurate method called static or vertical life table. Instead of studying a single cohort, the static life table compares population size of different cohorts throughout all range of ages at one point of time. In such cases, population ecologist frequently counts total number of individuals surviving at each age at a given time. In other words, they count the number of individuals of age 0 to 1 year, 1 to 2 year, and so on in the population. These counts can be used as if they were counts of survivors in a cohort, and the calculation is done in a similar way as that for a cohort life table. Static tables usually make two assumptions. 1. The population bears stable age structure. That means, in each age class, the proportion of individuals does not change from one generation to another generation, and 2. The population size remains more or less stationary. The construction of static life tables can also be done by estimating age of the individuals at the time of death in a given population. This life table is very useful for secretive large mammals such as moose from temperate regions where it is very difficult to do sampling of the living members. As the maximum mortality of large herbivores occurs during the winter season, an early spring survey of carcasses from predator kills and starvation can provide important information for constructing a life table for these animals. 13. 14. These two types of life table will remains identical only when the environment remains stable and does not change from one year to another and population remains at equilibrium. But generally birth rates and death rates of a population can vary from year to year, and as a result, there is large differences exist between these two forms of life table. These differences can be easily understood by analyzing the human population. For example, static life table for human that born in 1900 in the United States show what the survivorship would have been if the population continued to survive at the same rates observed in 1900. But the population did not retain the same rate because of the development of medical facilities as well as sanitation in last 117 years that has increased the survival rates and life expectancy of human population by more than 15 years. Static life table assumes stationary populations. Example 5. Static life table for the human female population of Canada, 2000. 9. Application of life table. Life tables can be used to understand the patterns and causes of mortality of the individual. 9. Application. (45 to 49 1,336,009). Static life table, time specific, stationary or vertical, is constructed on the basis of cross-section of population at a given specific time. The above static life table is calculated on the basis of census data and mortality data of Canada female population in 2006. This data can be used to calculate the mortality rate and other life table components by assuming that the population is stationary. 9. Application of life table. Life tables can be used to understand the patterns and causes of mortality of the individuals in a population. It can also be used to predict the growth or decline of future populations and also help in protecting the populations of endangered species. Another very important application of life table is the prediction of growth and decline of human populations. The growth or declining of the population of a country or given region 15 depends upon the number of children each person has and the age at which the individuals die. Moreover, it is also important to note that the growth or decline of a population also depends upon the age of the individuals at which they produce their offsprings. It is helpful in providing the effects of changing patterns of survival as well as reproductive events on the population dynamics. Life tables can also be used in the field of species conservation efforts. For instance, in the case of the loggerhead sea turtle of the southeastern United States, the population of loggerhead sea turtle is declining and mortality of their eggs and hatchlings is very high. These important information about sea turtle led conservation biologists to emphasize the protection of their nesting beaches. When these measures were not proved very effective in protecting the decline of sea turtle population, compiling and analyzing of their life table indicated the reducing mortality of older turtles could provide a greater probability of reversing the declining of these sea turtle population. Therefore, management efforts to protect these turtles shifted to convincing the fishermen to install the device of turtle exclusion on their nets to prevent the older sea turtles from drowning. Life tables can be used to calculate the average longevity of a population for determining the age composition of a population. It can also be used to indicate critical stages in the life cycle of a species at which mortality rate is very high to make the differences between species, for explaining success of same species and varied biotopes, for providing information of value in fish exploitation yield, as well as controlling of various agricultural and other pests. 6. Summary. A population can be defined as a group of individuals belonging to the same species. Some population either have defined boundary while others do not have well-demarcated boundary. Mortality is one of the important parameter which can explain the population change. A life table can be expressed as summary of age-specific mortality rates occurring in a group of individuals of a given population. 16. Life table construction is a simple method to follow births rate, deaths rate and reproduction in the given population. Life tables are categorized into 3 types: cohort life table that observe the survival of all individuals in a population from the time of birth to death, static life table analyze the total number of individuals living at each age group in a population, and dynamic composite life table is a composite of a number of animals that are marked for a period of years instead at the time of birth only. The first column X in a life table denotes age class and second column NX specify the total number of individuals surviving at the starting of each age. These 2 columns of the life table can be further used to find out several other life history parameters, such as the proportion survival LX proportion of individuals in a cohort dying at each age DX, and age-specific mortality rate QX, which is useful to find the age at which mortality is maximum. Static table makes assumptions that the population has a stable age structure and the population size is stationary. Study of life tables can be applied to understand the patterns of mortality, predicting the growth or decline of future populations, and also help in protecting the populations of endangered species. 2. Introduction. For a population, the surviving individuals are more significant for a population than the dead individuals. For a population, the surviving individuals are more significant for a population than the dead individuals. The death rate can be represented by survivorship curves or life tables, both of which provide an estimate of organisms surviving at various ages. The data obtained from a cohort life table can be represented as survivorship curve for a given population, which describes age-specific mortality through survivorship. The life tables and survivorship curves are derived from data obtained from a cohort of a species at given period of time under certain environmental conditions. In the previous module we studied about concept and construction of life table, and in this module we shall understand survivorship curve and also learn about fecundity table. 3. Survivorship curve. The life table can be represented on a semi-logarithmic paper in which the proportion of surviving individuals LX is plotted on the vertical axis y-axis on a logarithmic scale and age class X is plotted on the horizontal axis x-axis on the arithmetic scale. This representation is called a survivorship curve. 3. Figure point 1. Hypothetical survivorship curves on a semi-log plot, after R. Pearl, 1928. It can be categorized into three types. Figure 1, which can be used as a standard at which real-life survivorship curves of different organisms can be compared. It represents the different nature of survivors in different types of populations. 3.1 Highly convex curve. This type of curve, type 1, is the characteristic of the species in which the mortality rate of the population is low up to the end of the lifespan, old age. Thus, such species survive throughout their lifespan with low mortality rate in young and adult individuals. Many species of large animals such as deer, mountain sheep, man and small rotifers show such curves. Figure 2. 4. Figure point 2. Animals showing type 1 survivorship curve, pig on top and mountain sheep on bottom. Among various large-sized perennial plants, generally they die after reaching reproductive phase within a period of old age. A population which is characterized by a type 1 survivorship usually exhibit low birth rates. This is due to the fact that mostly all young individuals survive to perform reproductive event, and their high birth rate would lead to runaway population growth. 3.2 Diagonal curve. This type of curve, type 2, indicates an age-specific constant survivorship, i.e., a constant rate of mortality occurs at every age. Such a curve is characteristic of some animals such as hydra, gull, American robin, adult birds, rodents, and reptiles, as well as many perennial plants, etc. Exhibit this type of curve. Figure 3. 5. Figure point 3. Animals showing type 2 survivorship curve, rodent on top and American robin on bottom. 3.3 Highly concave curve. This type of curve, type E, is the characteristic of those species in which mortality rate is high during the early life stages. Some birds, oysters, shellfish, many insects, oak tree and fishes exhibit this type of survivorship curve. Figure 4. In oysters, higher mortality occurs due to high predation in the early age as well as adverse environmental conditions. In case of holometabolous insects, stair type of survivorship curve is obtained as different stages of insects in their life history exhibit great variation in their survival. 6. Figure point 4. Animals showing type 3 survivorship curve, fishes, insects, etc. Many birds, mice and rabbits show a slightly concave or sigmoid curve due to higher mortality in the young individuals. However, mortality is low when they reach to the adult stages. The population which is characterized by type 3 curve concave often required high birth rates to maintain the constant population size. The high birth rates make sure that sufficient numbers of young individuals survive to reach at reproductive age to ensure that the population can continues to maintain itself. Analyzing the shape of the survivorship curve is very important to find out the critical periods in the life history of the organisms. When the line of the survivorship curve becomes steeper, it denotes the increase in mortality rate of the individuals due to some environmental factors or some developmental effects on the population. For example, salmon fish produce several thousand eggs in its one-time reproductive event, but only few young ones are able to survive and reach the adult stage. Suppose, if only one egg out of 10,000 eggs survive to reach adult stage, it indicates that only 0.01% of the total invested energy is represented in the next generation. Most of the larger animals survived for several years and invest their energy. 7. In repeated reproductive events are considered as iteroparous, whereas many species of plants, insects and different microorganisms, after a single reproductive effort one or both individuals die, are considered as simulparous, which represents ultimate expenditure of energy to produce maximum number of eggs. Figure point 5. Survivorship curve of the song sparrow, Melaspisa melodia. The curve is typical of birds. Adapted from Johnston 1956. These three types of generalized survivorship curves can be used to compare the survival of a species. In fact, no population in the real world has a constant age-specific survival rate throughout the whole lifespan. Many species in their life history exhibit components of all three types of generalized survivorship curves. For example, in song sparrow, Melaspisa melodia, there is high mortality in juvenile stage, type 3 survivorship curve, and in adult stage it becomes linear, type 2 curve. Figure 5. The survivorship curve for the gray squirrel is presented in figure 6. 8. Figure point 6. Survivorship curve of gray squirrel, source, Smith & Smith, 2012. A slightly concave or sigmoid survivorship curve is characteristic of many birds, mice and rabbits. In these animals, the mortality rate is high during the younger stage but becomes lower and almost constant in the adult stage 1 year or older. In some holometabolous insects, i.e., insects with complete metamorphosis, such as butterflies, the survival rate differs in successive life history stages and the curve becomes the stair-step type survivorship curve. The initial, middle and final steep segments represent the egg population and short-lived adult stages, and the two middle flatter segments represent the larval and pupal stages, which exhibit less mortality. Some earthworm species also exhibit stair-step type survivorship curve. Lastly, crowding, high density, in certain populations such as black-tailed deer population may influence the shape of survivorship curve. The survivorship curve of human population is highly convex. This has become possible because of increased medical care, better hygiene, improved nutrition, and so on. The average lifespan of an Indian male has gone up from 30 years before 1947 to 58 years now. But maximum longevity has not gone up very much in any society. 4. Mortality curve. 9. 10. When mortality rate Qx of the life table is plotted against age x, it is called as mortality curve, figure 7. Mortality curve has two phases. The first phase is juvenile phase, where mortality rate is high, and second phase is post-juvenile phase, in which mortality rate first decreases with increase in age and then mortality rate increases with age after a period of low mortality. Usually, the mortality curve is J-shaped, and such curve indicates the rate of mortality through the slope of line is more as compared to survivorship curve. Figure point 7. Hypothetical mortality curve. Mortality curves for the life tables presented in Figure 8, gray squirrel. For the gray squirrel cohort, the curve consists of two parts. The first part is a juvenile phase where mortality rate is high, and second phase is a post-juvenile phase in which mortality rate decreases with increasing age until mortality reaches to some low point and then again it start increasing. Figure point 8. Mortality curves for a gray squirrel population. Source: Smith and Smith, 2012. XNXQXO 5300.71159.0.5280.4348.0.554. A life table demonstrates the mortality schedule of a population. The next step in the life table is to combine the mortality and reproduction to estimate the net population change. Alfred Lochta, 1925, derived one demographic parameter called intrinsic capacity of increase. Any population in the nature has its mean longevity, growth, mortality, and speed of development. These parameters are not constant for any species. Their mean value is determined by innate quality of the organisms and their surrounding environment. Due to fluctuation in environments such as winter and summer, the population cannot grow continuously. Population increases in favorable conditions and decreases in adverse condition. The organisms have great intrinsic capacity of increase. However, in nature there is always a balance in the population size. For example, female Helicoverpa armigera, a polyphagous pest, can produce 1,200–1,600 eggs in its lifetime. Even then there is a balance in the population size of this pest due to environment and biological agents. Therefore, in nature there is actual rate of change of population occurs in response to environment, age structure, and genetic composition. A standard convention in the study of populations is to express birth rates as births 1,000 population per unit time. This is calculated by dividing the number of births over given time period by the population size at the beginning and multiplying by 1,000. This will represent the crude birth rate, and this can be further improved by taking two important factors. First, in sexually reproducing population, only females give birth to young ones. Second, the number of birth given by female varies with age. Hence, birth rate can be expressed as the number of births per female of age X. As the size of population is increased due to reproduction by females, the age-specific birth rate can be further modified by determining mean number of females born to a female in each age group BX. Following given data for age-specific birth rates of gray squirrel population can be used to construct fecundity life table, Table 1. 11. XBX001223. Females of ages 2 and 3, the average number of female offspring produced. At the beginning, age 0, females did not produce any offsprings, so the value of BX is 0. At age 1, female produces an average of 2 female offsprings. Females of ages 2 and 3, the average number of female offspring produced BX increases to 3, and after that BX declines to 2 when female reaches to age 4. By the age of 5, females cannot produce offsprings, therefore, the value of BX is again reaches to 0. The summation of BX values of all age group provides an estimate of the average number of female offspring produced by female in her lifetime. It is called as gross reproductive rate. In the above example, the gross reproductive rate of gray squirrel population is 10. However, in this example, value assumes that the maximum survival of female squirrel is 5 years. Now, next step is to measure net reproductive rate that integrate the age-specific birth rate and the probability of survival of female to any specific age. After knowing the fertility schedule BX and proportion surviving LX, another important parameter, net reproductive rate R0, can be calculated. Net reproductive rate R0 equals number of female offspring produced in generation T plus 1 number of female offspring produced in generation T. Net reproductive rate is thus the multiplication rate per generation, and it can be calculated by multiplying LX and BX schedules and summing over all age groups. 12. If net reproductive rate is 1.0, the population is replacing itself, and if rate is below 1.0, the population will not replace itself. 6. Determination of net reproductive rate R0. The age-specific birth rate of gray squirrel population can be used for constructing a fecundity or fertility table. The fecundity table uses the survivorship column LX and aga-specific birth rates BX from the life table data. In the fecundity table, initially BX increases with the age, whereas proportion survivorship LX declines with each age group, Table 2. To adjust for mortality, each BX value is multiplied by its corresponding proportion survival LX values. The resulting value LXBX gives the average number of female offsprings produced in each age class, adjusted for survivorship. Table point 2. Fecundity table of gray squirrel population. XLXBXLXBX. 01.00.00. It represents that the survival of adult female. Similarly, for female of age group 2. Therefore, the value of Bx for 1-year-old female is 2, but when it is adjusted for survival Lx the value declines to 0.6. Similarly, for female of age group 2, the value of Bx is 3 and Lx Bx value declines to 0.45. It represents that the survival of adult females is poor. The values of Lx Bx are summed over all ages at which reproduction occurs. The values of Lx Bx are summed over all ages at which reproduction occurs that will give net reproductive rate. R0 equals sigma Lx Bx equals 1.40. 13. 14. Therefore the net reproductive rate of gray squirrel population is 1.4. If value of RO is greater than 1, females are more than replacing themselves. In this example, RO value of gray squirrel suggests a growing population of females. The difference between gross reproductive rate and net reproductive rates 10 and 1.4, respectively, reflects that only a small proportion of female gray squirrel will survive to the maximum age and produce 10 female offspring. As the value of R0 is a function of the agaspecific birth rate and survivorship, it is a product of life history characteristics, the reproduction time, allocation to reproduction, trade-off between the size and number of offspring produced and the level of parental care. Therefore, net reproductive rate R0 provides a means of evaluating individual, fitness, and population consequences of specific life history characteristics. The fecundity table of human females in the United States is presented in the Table 3. If human population of the United States continues to grow at these rates, it would multiply 1.039 times in each generation. Table point 3. Fecundity table for women. In the natural condition, environment is always fluctuating, sometimes favorable and sometimes unfavorable. When environmental conditions are favorable, there is increase in population size, and if conditions are adverse, population number declines. Therefore, in the natural state, a population shows the actual change that can vary from positive to negative due to change in social structure, age distribution, genetic composition, or change in environmental factor. An organism's innate capacity of increase Malthusian parameter depends on its fertility, longevity, and development speed. These processes are integrated with natality rate and mortality rate. The natality rate can be explained by birth schedule, egg or seed production of the population. The number of female offsprings produced by per female aged X to X plus 1 is considered as fertility schedule Bx. Usually females are counted for fertility schedule, and demographers considers only females as a population which further produces more females. Table point 4. The following given hypothetical data can be used to illustrate how the intrinsic capacity of increase R can be calculated from biological data. XLXBX LXBX Seven. Summary. A population can be defined as a group of individuals belonging to the same species. The surviving individuals are more significant for a population than the dead. The death rate can be represented by survivorship curves. Survivorship curves can be classified into 3 types. It represents the different nature of survivors in different types of populations. Type 1 or convex curve is present in the population of those species which exhibit low mortality rate up to the end of their life. Type 2 curve indicates an age-specific constant rate of mortality. Type 3 curve is the characteristic of those species in which mortality rate is high during the early life stages. These 3 types of generalized survivorship curves can be used to compare the survival of a species. When mortality rate is plotted against age, it is called as mortality curve. It has 2 phases, juvenile phase and post-juvenile phase. Any population in the nature has its mean longevity, growth, mortality, and speed of development. Due to fluctuation in environments such as winter and summer, the population cannot grow continuously. A standard convention in the study of populations is to express birth rates as births 1,000 population per unit time. After knowing the fertility schedule Bx and proportion surviving Lx, another important parameter, net reproductive rate R0 can be calculated. Net reproductive rate is thus the multiplication rate per generation. An organism's innate capacity of increase depends on its fertility, longevity, and development speed. These processes are integrated with natality rate and mortality rate. The length of a generation G can be explained as the mean period between the birth of parents and the production of the offspring. 17.